Thank you. I'd like to express my sincere thanks to the John Dirks Canada Gairdner Foundation for the 2021 Global Health Award given to my colleague Iguan and myself. We recognize this not just as an award to the two of us, but as a recognition of our whole research group at the University of Hong Kong. What I'd like to do in the next few minutes is to share with you my personal journey in research. And I've entitled it From Pasteur to SARS and Beyond. When I was a 14-year-old high school kid, I read this book on the life of Louis Pasteur. And I was inspired by the fact that not only was he an eminent organic chemist, but he went on to develop the germ theory of disease, establish the scientific basis of vaccination, and translate these basic findings to application. And finally, he was also very active at disseminating his knowledge by establishing a network of Pasteur laboratories worldwide. And come forward a number of decades, I was fortunate to become one of the directors of one of these Pasteur network laboratories, the Pasteur Research Pole at the University of Hong Kong. But let me go back to my roots in Sri Lanka. After graduating from medical school, I was still driven by the desire to pursue a career in science. And in this, I was inspired by the then professor of microbiology at my alma mater, the University of Peradeniya, Chabi Arasakulratne. He was one of the few people who was actively carrying out good quality research in a very resource limited setting. And he showed me that it is possible to do research in a place like Sri Lanka. So after my postgraduate training at the University of Oxford, I returned to the University of Peradeniya to set up a cell culture and virology laboratory. And this, of course, was challenging because I was setting this laboratory from scratch. There was no virology or cell culture in the University of Peradeniya, or indeed in very few places in Sri Lanka. But nevertheless, we were able to carry out some very productive research activities, train 5 graduate students, and made the first monoclonal antibodies to Plasmodium vivax. And we use them in collaboration with a parasitologist colleague of mine to identify targets of transmission blocking immunity, and also studies on mosquito-borne viruses. But one of the studies I want to highlight is at that time, the longest and the largest river in Sri Lanka was being dammed at multiple sites for generation of hydropower and to irrigate land for agriculture. So since we were interested in mosquito-borne viruses, together with an entomologist colleague of mine, we decided to set up a surveillance site at this irrigation program and development program from the time of its forested state right through the changes that took place, forest clearing, irrigation, development of agriculture, and human settlement. And our hypothesis was that we would uncover emerging mosquito-borne viruses in this region. And as often happens, what happened was an outbreak not in the region that we were studying, but in a different part of the country, the city of Anuradhapura, which was not undergoing any major ecological change. So nevertheless, since we had the tools all set up, we studied this outbreak in detail, and we identified the mosquito species that carried the virus, investigated the patients with the disease, and the virus itself. And there was really no reason why, out of the blue, basically, there were Japanese encephalitis, which up till then, there had been a dozen or so cases every year in the country. Now, why we would have 500 cases in one hospital in the country. And what turned out to be the case was that just a year or two before that, the local provincial authorities, with the very best of intentions, had decided to introduce smallholder pig husbandry to these rice farmers. They gave each of these rice farmers 10 pigs to have in their backyards. Now, given the situation that this was an area where the ecology was such that the mosquito vectors were present at very high density, and of course the virus is present in the environment in wild water birds, the introduction of the amplifying host, the pig, was what led the spark to this huge explosion of this outbreak. So this really was an early lesson to me that how important it is that we have to consider all aspects of our environment in considering emerging infectious diseases. After that, I went on to risk assess which parts of the country were at high risk of Japanese encephalitis and where the risk was minimal and where pig husbandry can be safely carried out. So this brings me to this concept of One Health, which is the optimal health for peoples, animals, and the environment, and which requires the close interaction of human health, animal health, and environmental sciences. And I think that early experience with the Japanese encephalitis outbreak really was fundamental in driving that message home to me, although at that time, this concept of One Health was not really widely talked about or was very popular at that point in time. Fast forward a decade or so, I took up appointment at the University of Hong Kong, and one of my tasks was to set up a diagnostic clinical virology lab at the Queen Mary Hospital, which is the teaching hospital of the University of Hong Kong. This we did, and within 2 years, we were then confronted with the H5N1 avian flu outbreak. There were 18 human cases with 6 deaths of a completely novel avian influenza virus. And this was the first time that avian influenza virus had directly jumped to humans, causing severe disease and death. The source of the outbreak, source of human infection, was identified to be the live poultry markets, and the culling of all poultry in Hong Kong led to the termination of that particular outbreak. But after that outbreak was over, I was interested, together with my colleague Yi Guan, and mentored by Dr. Robert Webster at the St. Jude Children's Research Hospital, who in fact mentored both Yi Guan and myself, to set up systematic surveillance in these live poultry markets and also in wild birds and in pigs, to look for the precursors of the H5N1 virus. And indeed, we found that with imported poultry, we could find evidence of the H5N1 virus, and also demonstrated that the virus was continuing to evolve. And I'm sure my colleague Yi Guan has explained this in much greater detail in his talk. But by studying the poultry marketing system, we were able to establish that what was occurring was that because there were multiple sources of poultry coming in from multiple farms through a single wholesale poultry market, and then being distributed into smaller retail poultry markets, that once a virus gets into one of these retail poultry markets, it essentially never leaves, because you always have fresh, naive poultry being introduced into this setting, and poultry being removed for slaughter. And this gives the virus an ample opportunity to continue to infect new incoming poultry. And furthermore, we also demonstrated that when we had outbreaks in poultry farms, what was happening was the introduction of the virus from these retail poultry markets to poultry farms through the movement of cages and individuals carrying a contaminated virus back into these poultry farms. So this allowed the introduction of a number of intervention measures to reduce risk. And I will just show you the impact of some of these. in order to break the maintenance of the virus in these live poultry markets, we realized that the only way this could be achieved was by emptying the market completely, free of poultry. And then we used one whole rest day per month in these poultry markets, then 2 rest days a month, and then finally made sure that live poultry were only sold during the day and the market was emptied during the night. And what you can see here is 12 years of systematic surveillance in these poultry markets. And the data here is from a low pathogenic influenza virus, H9N2. And you can see the impact of these interventions, particularly banning of live poultry holding overnight, dramatically reduces virus persistence and activity in these markets, and thereby reduces zoonotic risk. So with these interventions, we were able to prevent any further cases of severe avian influenza in Hong Kong after that 1997 outbreak. But unfortunately, the reports that we were making public through reporting to WHO, OIE, and also in research publications was not taken heed of by other neighboring countries. And in 2004, suddenly, many countries and territories woke up to the fact that they were having H5N1 outbreaks in their poultry and sometimes affecting humans as well. So one might wonder whether if this whole region had taken the same surveillance and proactive measures that we had taken in Hong Kong, whether the extent of this H5N1 outbreak that took place subsequently might have been mitigated. Another area that I was interested in was the severe disease that this H5N1 virus caused. Human infections was associated with about 50% fatality. And we were able to show that the H5N1 virus elicits a very different innate immune response activation profile compared to seasonal influenza viruses. So we believe that this dysregulation of innate immunity was part of the pathogenesis of the severe disease H5N1 virus caused. In collaboration with my colleague Michael Chan, we developed experimental models to study the alveolar fluid leak problems that occur in the lung in these patients. And we were able to show that what happens is that when H5N1 virus infects alveolar epithelial cells, these are the air sacs in the lungs, they release soluble mediators which downregulate ion channels which are found in these alveolar epithelial cells. These are, if you like, the pumps that remove fluid that is seeping in to these air sacs. And if these pumps are inactivated, you have accumulation of fluid in the alveolar spaces, leading to acute lung injury. And we were also able to show that this damage can be reversed, for example, using cell therapy such as mesenchymal stromal cell therapy. And now we are using these models to look for other small molecule therapies that might reverse this damage to the ion channels that we have observed. Now, if you go back a bit in time, back to 2003 again, we had heard of an unusual pneumonia that was taking place in Guangdong, the adjoining province to Hong Kong. And we set up surveillance with the hospital authority in Hong Kong. And we were able to quickly identify a possible virus that might be associated with these cases and showed that this virus was the cause of SARS. And also that this was a novel coronavirus. So by obtaining partial genomic sequence of the virus, we could show it was a new coronavirus. And this also allowed us to develop diagnostic tests for the virus, which we gradually rolled out in use within a week of identifying the virus itself. Now, this is not normally what someone would do to roll out diagnostic tests so quickly, but given the emergence situation, we had to do this. In other words, to build the ship as we sailed, if you like, evaluating the test as we were using it in the field, and we were able to successfully do this. We were then able to identify a number of clinical and virological characteristics of this infection. We were able to show that it was not just an infection of the respiratory tract, it was also infecting the gastrointestinal tract. The virus was present in the stool, and in fact, many of these patients had diarrhea. And at least in one instance, fecal contamination was responsible for a huge outbreak in Hong Kong. We were also able to show that quantitatively, the amount of virus in the upper respiratory tract in these patients was quite low in the early stages of infection, and it peaked at the end of the first week. So we therefore supposed that the infectivity of patients early in the infection was quite low, and indeed that proved to be the case. Epidemiological data also showed that transmission was less likely in the first 4 to 5 days after onset of illness. And there was also another unusual feature of this disease, because many respiratory viruses essentially have this iceberg phenomenon where most infections are mild or asymptomatic and not recognized, only a small proportion of them being severe. With SARS, it was exactly the opposite, with most cases being clinically overt and quite severe. So these two factors taken together, the fact that transmission did not take place very much in the first few days, and that most of the cases did present to medical attention, made it possible for patients to be detected, isolated early, and for transmission to be interrupted. And that's how the global outbreak, in fact, was brought under control. Now, as I said, with other respiratory viruses like influenza, virus infection and transmission takes place from the time or even just before the onset of clinical symptoms. And in 2020, with the emergence of COVID-19, in collaboration with colleagues in southern China, we investigated a cohort of patients, and by early February of 2020, we were able to report that the virus load profile in COVID-19 was very different to that in SARS, and we said that the transmission pattern is more likely to be that of influenza rather than that similar to SARS of 2003, so that the strategies that we used to successfully contain SARS in 2003 were probably not going to be able to interrupt transmission of COVID-19. And unfortunately, that proved to be true. Now, one of the features of the SARS outbreak was the very close collaboration of multiple laboratories across the world working together under the coordination of WHO. We, on a daily basis, in the case of Hong Kong, it was evening or night, when we had daily teleconferences sharing progress that particular day, and we were sharing information in real time, which was extremely important in the overall response to that outbreak. And some of the people that were involved in these calls, who I got to know, included the late Don Lowe, who was head of microbiology at Mount Sinai in Toronto, and Theresa Tan, who is now the Chief Medical Officer of Health for Canada. Now, the question of how SARS emerged, I'm sure my colleague Yi Guan would have explained. This was work done by him, where he showed that the live game animal markets in southern China was where the jump of the virus took place from animals to humans, and the closure of those markets almost certainly prevented the re-emergence of SARS from that reservoir after the human outbreak would have been contained. And subsequently, it was shown, we ourselves very early showed that bats do carry coronaviruses, and then subsequently others showed that SARS-like viruses were found in these Chinese horseshoe bats. But it took almost 10 years to find the near precursor to the 2003 SARS coronavirus. With the emergence of COVID, of course, our laboratory was very involved in responding to that outbreak. We developed diagnostic tests very early, and one of the earliest diagnostic protocols that we saw that was on the WHO website came from our lab. And we were, on request, provided diagnostic reagents to over 170 labs in 77 countries in those early weeks. before commercial diagnostic tests became available. And a number of countries confirmed their first cases through these reagents. We had been very interested in trying to understand the modes of transmission of respiratory viruses, particularly influenza. But some of the patients that we were studying were having other respiratory viruses, including the seasonal coronaviruses. This is not COVID-19, but this is the endemic seasonal coronaviruses that infect humans. And we were able to show that these viruses were found in respiratory droplets, both large respiratory droplets and very fine airborne particles. And importantly, we found that when the patients wore a surgical mask, the dissemination of these airborne droplets from the respiratory tract of these individuals was essentially reduced very dramatically. And this was one of the scientific basis for the recommendation of universal surgical masking, particularly once it became known that asymptomatic people and pre-symptomatic individuals, that is people who got infected and in the day or so before developing symptoms, could transmit disease. So the basis of wearing a surgical mask, reducing the spread of these respiratory droplets from infected patients. Now, one of the other things that I have been interested in is the MERS coronavirus, which, as you know, emerged and was first detected in the Middle East. But one of the surprising findings in the case of MERS is that over 75% of MERS coronavirus infected camels, which is the source of human infection, are actually found in Africa. But human MERS has not been reported, MERS disease has not been reported from Africa. And we did studies in a number of countries in Africa in collaboration with local veterinary and human health authorities to show that the virus carriage in these camels in the African continent was very similar to that in the Arabian Peninsula, where cases were being reported. So we have more recently gone on to show that although these camel workers have no detectable antibody or very little detectable antibody to show evidence of infection, that using T-cell responses, that we can in fact show that about 1/3 of these people do have evidence of past infection. So that infection is taking place in Africa. We have gone on to show that the MERS coronaviruses in Africa are genetically distinct from those in the Arabian Peninsula and may have reduced virulence or ability to cause human disease than those in the Arabian Peninsula. But we also have shown that one of the determinants of this apparent difference in virulence is the spike protein of the virus, and these viruses differ from the Arabian Peninsula viruses in just a few amino acids. So these viruses from Africa may pose a major public health threat, and we point out the need for enhanced surveillance of MERS coronavirus at the animal-human interface in dromedary camel herding regions in Africa. So if I may just summarize, I think I have tried to highlight the importance of the One Health approach, that is looking at human, animal, and environmental health in an integrated fashion in order to respond to these emerging infectious disease threats. The importance of surveillance and risk assessment and proactive intervention so that we can understand the risks, understand the pathways of emergence, and then take interventions to reduce the risk at source, if you like. And I've given you the examples of avian flu and SARS in 2003. So I would like to highlight the fact that surveillance and risk assessment is essential for pandemic preparedness. It is not dangerous science, as some people are now trying to say it is. And I would like to highlight that, unfortunately, future pandemics are inevitable, and COVID-19 may not necessarily be the most severe pandemic that the human species has to face. So I think it is important that we learn the lessons from COVID-19 and be much better prepared for the future. And my last slide, I would like to highlight and to think beyond pandemics. I think COVID-19 highlighted the limits of human hubris. I'd like to quote this sentence from René Dubos, who said this in the 1950s, at a time when people were thinking that with the development of antibiotics and vaccines, that infectious diseases were well controlled. He said that at some unpredictable time and in some unforeseeable manner, nature will strike back. And I think this is what we are seeing with a number of emerging infectious disease outbreaks, and of course, culminating in the COVID-19 outbreak that we see right now. And it is not just pandemics that we have to be worried about. Our human activities are really leading to a whole range of threats, including climate change, environmental degradation, environmental pollution, and we risk rupturing the limits of planetary sustainability. And I think it behoves us to use COVID-19 to mobilize public concern and public action so that we change our behavior so that we can live within the sustainable limits of our planet Earth. With that, I will end, and I'd just like to acknowledge many people who are involved in this work. This is a picture that was taken soon after the discovery of SARS in 2003, and it shows some of the people involved. Leo Poon, who did some of the initial sequencing, Dr. K.H. Chan, who was fundamentally important in culturing the virus, and then Don Nichols, who did electron microscopy, Yi Guan, of course, who is my co-recipient of the Gairdner Prize, and K.Y. Yuan, who was involved in this work as well. I mentioned the important role of Robert Webster, who was a mentor both to Yi Guan and to myself. Ken Shortridge, Gabriel Leong, who is the director of the School of Public Health and now the dean of our Faculty of Medicine. And of course, in subsequent years, many other people that I have had the good fortune to work with. And of course, a lot of this work was done in collaboration with multiple authorities, the Department of Health, the Department of Agriculture, and the Department of Food and Environmental Hygiene. I thank you.